Hello everyone, I'm Mr. Furlong, and today we're going to talk about how that first cell formed. Remember that we are going to be taking a look at the origins of life through this process of chemical evolution. Now we talked about three other theories, but chemical evolution is the one that falls within the realm of science. So for instance, we can make hypotheses, we can test those hypotheses, we can revise our hypothesis based on evidence that we've collected. This is why we're specifically looking at chemical evolution. Now remember what the early Earth was like. Very different from today. The Earth began to cool, all that water vapor started to condense into liquid water, the gases that were present in the early atmosphere started mixing and churning with the water, forming that primordial soup. So how can all of that then get us to a living cell? Well, there's many different theories on this, and we're not going to talk about all the different theories. We're going to simply take a look at what are the steps that they all agree upon that would have to have occurred in order for those first living cells to have come about. And so this first step is we have to make the simple organic compounds. All living things are made up of organic compounds, but there weren't any present in the early oceans. So how did we get things like glucose, fatty acids, glycerol, amino acids, nucleotides? A famous experiment done in the 1950s is the Miller-Urey experiment. We're going to ask just that kind of a question. Could it be possible for organic compounds to form given the conditions of the early Earth? So they set up an experiment where they simulated the conditions of the early Earth. This is a drawing of what it looked like. So a few things to note here. They started with water, and that was just fresh water. Remember, the oceans were fresh water. They added some heat to that water, which created steam. So now we have some water vapor that made its way into this larger bulb where they put different types of gases. So they put in things like carbon dioxide and ammonia and methane gas, and they gave it a little spark of electricity every once in a while. So this was simulating the gases in the atmosphere, but it was also simulating the fact that there's lightning strikes going on. And it cycled around to the point where it cooled. The water vapor became liquid water again, which would then be reheated, and this whole thing continued over and over again. So when they came back the next day, what they found was that this water, which was clear to begin with, was kind of like this dark red color. And when they tested it, they found amino acids were in that water. Now this was huge news. In just 24 hours they were able to make the building blocks of proteins, and all living things are made up of proteins. So like any good scientist, they published their papers, other scientists would read about it, and they said, well, wait a minute here, you put these gases in, we think that maybe these gases are present. So they repeated this, and they repeated it over and over and over again. And they ended up making other types of organic molecules. So what's important about this experiment is that they showed that organic compounds could have been made this way. It's not proving that organic compounds form this way. It's simply showing that it certainly is possible. If we can do this in the laboratory, maybe 4.5 billion years ago, the same types of things were going on. Now, as I mentioned before, they repeated this over and over and over again. What they ended up making were all 20 amino acids. They made the 5 different bases of the nucleotides. They made phosphates and the sugars ribose and deoxyribose. So all the basic building blocks were made. In fact, they did this so many times that even today they are taking vials of this water that was made and they're still testing them. Now all of these things would have been present in the primordial soup. So now if the basic building blocks, all these monomers that we talked about way back at the beginning of the year, if all these things were present in those early oceans, maybe then they can start to form larger organic compounds. Certainly we're not anywhere near something being considered alive at this point. The second step is the formation of complex organic compounds. So all we have are these simple building blocks. They need to now form into things like proteins, into polysaccharides, into lipids, and so on. So another scientist, a guy by the name of Oparin, discovered that organic molecules, they can arrange themselves together if they're in a particular pH. He called these molecules coacervates. Now here's a picture of some of the coacervates that he made. But what he was able to show was that, you know what, it is possible for molecules to begin to organize themselves. Many times we hear that molecules, of course, they want to go from a state of order to disorder, and in evolution, that's going in the opposite direction. But if you provide energy, you certainly can go from a state of disorder to a state of order. Now these are pretty simple things to make. In fact, we're going to be making these in class, and we'll be able to see them forming under the microscope. The third step is that organic compounds had to become protected against the environment. I mean, this is a very volatile place. They didn't have ozone protection. So ultraviolet rays is coming down. Anytime DNA wants to form, it's going to be destroyed. So these molecules have to be protected somehow. We don't know exactly how this happened. There's, again, a variety of theories on this, but somehow some type of pre-cell had to have been made. So a scientist by the name of Sidney Fox discovered that proteins, when you heat them, can form hollow spheres, and he called those microspheres. Remember, the water is pretty hot. It's still close to boiling. Now what Fox had shown is that this may have allowed for organic molecules to become separated from the external environment and then protect them from those harmful UV rays. The fourth step is the evolution of growth, metabolism, and reproduction. Now this is a big leap from going from a pre-cell to a living cell. And there's lots of different ideas on how and really where this had taken place. But essentially, molecules need to be able to reproduce. The only molecules that can do this are DNA and RNA. Now, since RNA is a simpler molecule than DNA, it's thought that RNA probably evolved first. RNA, of course, as we know, has the ability to arrange amino acids into proteins. So if they can reproduce and they can grow and they're maybe able to start to use energy, they now can be considered a living cell. Scientists have had a hard time, though, getting cells to do this. Currently, there is a study that's going on at MIT in Boston, and they were having the same sort of trouble. How do we get these molecules to start to reproduce? And all of a sudden, one of the graduate students said, hey, you know what? How about these clays? These clays are set up in such a way that molecules attach to them. And when they started adding these clays, what they noticed is that all of a sudden these molecules started putting themselves in order and, in essence, kind of started to organize themselves. So what were these first life forms probably like? Well, we want to use this idea that simpler things probably evolved before more complex things. So they're probably prokaryotic. In other words, they did not have a nucleus versus a eukaryotic cell. Most likely they were unicellular, so they were only made up of one cell versus a multicellular organism. They were also anaerobic, which means they had to live without oxygen. Remember, there was no oxygen present in the early atmosphere. They were also heterotrophs, so they had to obtain food from their environment. Now remember, an autotroph would have to be able to carry out two processes, photosynthesis and cell respiration. Heterotrophs would only have to carry out cell respiration. Now, is it possible that an organism can meet all of these characteristics? Certainly, we have organisms today that live in just this type of environment. Here we see some organisms that live in the bottom of the ocean. And when we look at the bacteria down there, they're all unicellular, they're all anaerobic, and they're all heterotrophs. It's estimated that life formed about 3.5 billion years ago. And this is the oldest fossil ever discovered. Now, this was found in something called a stromatolite. Here is a fossilized stromatolite. And what you want to focus on are these things right here. Now, notice they form in layers. And there's another one right over here. But these are made up of colonies of bacteria, of prokaryotes that had fossilized. And now we can see what those organisms may have looked like. We might be looking at the very first species on the planet, or LUCA, the last universal common ancestor. What do stromatolites look like today? Well, these are living stromatolites. These are found in Australia. And so these structures that you see here are what has been fossilized there. But then how can we get more complex cells? How can we get eukaryotic cells? Well, there is a theory called the endosymbiotic theory that says that a prokaryotic cell, which is a bacteria, is then housed inside of another prokaryotic cell or its host. So let's say here we have a prokaryotic host. Here's a couple different types of bacteria. We have some bacteria are photosynthetic. We have photosynthetic bacteria today, some are aerobic. And the endosymbiotic theory says that some of these bacteria, like these aerobic bacteria, may have started living inside of a prokaryotic host. And now we have this mitochondria containing prokaryote. Well, this could have continued where now we can see photosynthetic bacteria inside of this prokaryotic host developing what might become plant cells. What this arrow is representing is a spirochete. That is another type of prokaryote that's similar to a flagella. And eventually maybe fungal cells had formed. We can have the formation of animal cells then, all coming from other prokaryotic cells living inside of another prokaryotic cell. And then eventually all of these eukaryotic cells started to evolve. Of course, why would cells want to start living inside of another cell? Right? Natural selection is going to have to take place here, right? I mean, there's got to be a benefit. Well, so here's some of the benefits. For the bacteria, like the photosynthetic and the aerobic bacteria, they're now protected from their environment. Remember, all these UV rays coming in. The prokaryotic host, on the other hand, well, they can get things like food from the photosynthetic bacteria, or they can get energy from the aerobic bacteria. So both types of bacteria are benefiting from this arrangement. This all sounds great, but what sort of evidence do we have to suggest this? Well, we know that all cells have a complete set of DNA in the nucleus. However, chloroplasts and mitochondria have their own set of DNA, and they still use that DNA. When plant cells need to make more chloroplasts, they use the chloroplast DNA. When we need to make more mitochondria, the mitochondria uses its own DNA. The question is, why do chloroplasts and mitochondria have their own set of DNA? No other organelle does. The ER doesn't have it, right? The ribosomes don't have it, just the chloroplasts and the mitochondria. Maybe it's because they were their own individual organisms at some point 2.5 billion years ago. Lots of interesting theories and ideas that are going around, and this whole idea of how the first cells form is constantly changing. We're always getting new data, new ideas. We're going to be doing more activities dealing with this in class, and I can't wait to see you then.